If we had a jackhammer tool, Kira would like a piece of this rock.
Huh. All right. Perched on a dime. Go ahead and push on in there, please, Jonathan.
So Jess, can you collect while you're in the water column, or you have to settle down?
It's hard to collect when you're in the water column like this because you have some autos that bounce you around. So right now we're actually just perched on the rock face a little bit.
The corner you can see on bubble cam.
Yeah.
Over here.
Got it.
Holding on there.
All right, go ahead.
Barely perched.
Yeah, we get some stills of that. That's perfect. That looks different.
All right, go ahead and come wide there, please. Thank you. How much of a sponge would you like?
That's the wrong question to ask me.
Roger that. Go ahead and push on in there a little bit, please, Jonathan. That's great there. Huh, it's an interesting grab, this one. Come a little wide, please. A little wider. Thank you.
Solid view and bubble.
Raj, thank you.
You can see the crinoid in the upper left.
Beautiful. Contrast to that yellow one that was collected earlier.
Just thought I'd give you a little extra challenge this, since it's almost the end of the cruise.
Roger that.
Do you think it's a glass sponge, Paul, or a demo sponge?
Glass.
Oh,
it's
that's not what you want.
It's peeling nicely.
That might be what you want.
That was intentional. Let's just say that.
Yep.
Oh, we can get you a little bit more this way, I suppose.
Got a potential genus as Poecilastra.
Poecilastra.
Or the fringe shelf sponge. It's very close to what
it's a demo
you're
sponge,
calling
Greg.
it.
Yeah, good, even better.
Yeah, I was wondering if the fringe was the normal
nice.
glass spicules, but I guess they're not. It could be spicules still, but it just looked a little demo spongy from the surface.
Full wide, please.
Yeah, good call.
I'm assuming this is going in the front here too?
Yes, please.
And
All right.
outboard.
Go ahead there, Jake.
Popping out.
Thank you.
Hopefully it sinks on in there. Looks like it is.
Didn't even need to open my jaws.
Awesome.
Works out
Great.
nicely.
Perfect.
Well done.
The crinoid didn't swim away.
That's going to be sample NA124-181, and it was stored in the forward bio box number 5 red.
Copy.
So what else would you guys like from here?
I think we're good.
We
Thanks.
good?
Unless
Roger.
Paul sees something else he wants.
I think I'm ready to roll,
We got a trail of breadcrumbs
unless
in our sonar.
we can just scan a little as we pass over the rest of this rock.
There's an amazing feature ahead of us once we get up slope, so I think we've got
Roger that.
some cool things to look at.
All right, so continue on upslope and trying to make it to that.
Yeah, I think we'll want to stop before we get to the slope to finish the push coring.
Sure thing.
So
Would you want to do that here or wait a little bit?
do we have a little bit of a dip ahead of us, or is it just a
It looks
flat, very
pretty flat here.
Yeah, let's do it in that little zone there. Thanks,
Okay, yeah.
Renee.
Okay, we'll still be going 050.
050, Reg.
I'll call it in because it'll take a
Are you
little bit.
full wide on Argus?
Roger that, thank you.
Oh, thank you.
I think you're
Bridge
closer than
now.
you were.
Yeah.
— 100 meters, 050. Thank you.
I'll work my way over here for you, Jake. I can go to 050.
050.
Looks like there's a bunch of these fringe shelf sponges all along the rock here.
That trash.
Yeah, we just picked the most difficult one for just to collect.
It's like some tinfoil.
Those are quite interesting, though.
Yeah. It kind of looked like the fungi you see on trees, or those
Yeah.
plate mushrooms. Instead, we're under the sea at
Yeah.
1,600 meters.
Yeah.
There's another green bomber
Oh yeah,
and another.
they're loving this sediment. This whole area. Oh, another one of those big fringed shelf sponges.
That's chock-a-block full of biology. Look at that.
Wow. We can see it's only on one side of the rock, which could say something about the current here and where these creatures are collecting their food from the water column.
Is that a snailfish?
Another snailfish.
Jonathan, you need a time lapse of me, Jess, and Jake turning our heads in like different directions, looking at all the different screens as we're moving. Yeah. Sometimes we're in sync, but usually we're all looking at different things. Everything's covered.
Could these possibly more of those Faria sponges that we've seen? A bit more translucent looking sponges.
thinking about doing the cores here.
Yeah, we'll stop in here.
A few more benzodides. They're so purple.
They're kind of cute.
Lisa, if you're listening, how many push cores would you like? 3?
I'm curious how deep these push cores are going to be here. We can try it out for sure, but
If we wait a little bit, yeah, there's a little flat section ahead, I think, where there hopefully won't be rock before we start going up slope. Yeah,
Rog.
just in front of the ship position.
Yeah.
Roger that.
So Lisa needs 3, but we could also use more too.
Sure.
So I think there are 1, 2, 3, 4, 5. Oh, sorry, 1, 2, yeah, 5 remaining.
I think before we
So-
touch down where we want to do a push core, we'll also want to do a water sample
Yes,
as well.
yes,
Just to make you aware of that, Jess.
Yep.
Roger that.
Thank you, Caitlin.
So I know there was a really nice explanation of push cores earlier, but if you've tuned in a little bit later in the day, what we're looking for now is a good place. You know, Paul has his microbe glasses on, scanning for the best microbes in these sediments, but really what we're trying to do is just find an area that looks like we can get a deep enough push core, which is essentially pushing a tube into the sediment. It's like taking a slice of cake, and we're interested in separating out all the different sections of the core, like you'd layer like a layered cake, and we're really interested in looking at the microbial diversity, you know, all the different bacteria and microbes that live in each of those subsections. And as you get deeper in the sediment, you can imagine the oxygen decreases. You might see increases in other elements or sediment composition that could affect the community at each of those really, really tiny layers. And so these push cores are a really great way to get a sampling of the microbial diversity across the sediments. And so we're looking at the microbial community and who might be there and what are they doing. And Lisa's, we're also collecting some samples for her analyses, and they take and they section the core similar to how we do, and they set them through different size sieves, can never say that, different sized mesh screens separating out microfauna, meiofauna, megafauna, I'm probably forgetting a fauna, but different size classes of creatures that will really help, you know, things that, you know, with a microscope or a really keen eye might be able to see and can quantify perhaps the different stratification of what lives in each of the layers of the sediments.
I wonder if some of these returns that we're seeing here are just actually the really hard substrate underneath a thin layer of the sediment.
I suspect that those are rocks.
Yeah, I'm wondering if they're like, because when I turn my heading this way,
Right.
there should be some like all around us here.
That's like 10 meters out. See what we see. There's a little bit.
There's a little bits, I suppose.
Interesting. If there's rocks really shallow, that would affect our ability
I
to
don't
get
think
a full
that
core.
the 600 kilohertz would penetrate very far is the only reason that I
Yeah.
suspect not, but could be a bit.
Well, we'll soon find out.
Yeah. I did crank the gain up though, so he's bolder.
Oh, there's one.
There's
Couple.
a rock.
Which I can turn down. It was just that sediment wasn't showing up very well, and I wanted to.
No worries.
Yeah, thanks, Caitlin, for that explanation. If anyone's wondering how we estimate microbial diversity, it used to be you would culture bacteria and identify what you could culture, but we now know that you generally only culture a very small percentage of the bacteria that are present in a sample with the methods that are commonly used in the laboratory. And so instead we extract DNA from the sediment and we can assess bacterial diversity very, very effectively by sequencing certain marker
Is
genes.
that a line down there? I think that's, or is that biological?
Can't tell.
Can't tell.
Go ahead and fish on in there, please, Jonathan. I think it's biological. Full wide, please. Ah, no, actually, no. Just
I
kidding.
think you're right. I think it's a line.
I think it's a line.
Yeah, wrapped around the rock.
Yeah.
Very thin.
More of those plate sponges.
I think I went to the bottom side of that rock there.
Yeah.
There's another crinoid.
Beautiful yellow color. Nice contrast on that Faria sponge.
Quite a little a bit of layback here.
Raj.
So when we do want to stop, we'll just have to be patient about that.
Anticipate the swing.
Yeah.
All right, Jake. I'll get out in front of you again now.
Roger.
Maybe actually we should just hold position as we get this sandy bit. Let's see what sonar says.
After that.
I'm seeing so many pelagic worms. It's amazing. Lots of green bombers. It's kind of easier to see their shadow in the sediment than their actual bodies. But when you do see them, they swim in this gorgeous sinusoidal motion.
So we're holding the ship and we're just anticipating that we'll be in the flattest bit for this sediment in this area.
Yeah.
When we settle out before we start to get too steep.
Yeah, we better choose something soon.
Yeah.
To try. It's a lot of shells there.
Yeah.
Yeah.
Don't see any live ones.
Yeah.
Maybe there's an octopus around.
Really big octopus.
Could be.
Sorry.
Wow. Yeah.
When we see shells like that, we often wonder about a seep.
Oh, really?
But yeah, I think they were the
course.
ace. I think they're the acesta bivalves, though.
A little shelf.
Coming up.
This
Quite
is
a
a really
boulder.
cool rock face here. There's more
Yeah.
of those.
You can see how much
The
it's
brachiopod.
shadowing in the
And
sonar.
her, yeah.
Interesting.
Big. Came from above. Perhaps it came from this feature we're trying to eventually get to.
Can you explain why clams might be associated with seeps?
Yeah, there's a couple of kinds of clams and also a kind of mussel that have adapted to methane seeps and hydrothermal vents. which produce a lot of hydrogen sulfide, which is a toxic substance. It binds to our hemoglobin, for instance, and so you would quickly suffocate in the presence of a lot of hydrogen sulfide. But it's a good energy source, so there are certain bacteria that can use hydrogen sulfide, and these clams and mussels have adapted to have these bacteria live in their gills, and they basically farm them there and live off the bacteria and can grow to quite a substantial size. And we don't normally see big bivalves in the deep sea because there's just not a lot of food for them to filter feed, but the mussels can get by with a combination of filter feeding and bacteria, and the clams actually seem to just get up to well over a foot long, living only off the bacteria that they're farming in their gills. So when we see their distinctive shells, we often have evidence that there's hydrogen sulfide nearby, which is an indication of a seep or a vent, but we're not expecting any vents around here.
Awesome.
Is there certain types of corals and or sponges we should keep our eye out for?
Ah, someone wants a prize. Yeah, absolutely. So
Can we zoom on that branching structure there? Is that a crinoid or a coral?
Oh, yeah, sure thing. Stand by.
Good eye. That was very camouflaged.
I got the prize. If it's a coral. If it's a coral.
If it's a coral. Whoa.
I think it's a black coral, maybe. Steve will help us.
there you go.
Yeah, I'm betting it's a black coral.
Good eye, Greg.
All right, go
Good
ahead,
eye.
Jonathan.
Oh no, now I'm betting it's a crinoid.
Yeah, looks like a
That's
stalked
a-
crinoid.
Oh,
Docked crinoid.
Looks like a
no, it's a
coral.
coral.
Oh,
It's a
Yeah.
coral.
Is it?
Yeah.
Coral.
Black coral.
Interesting.
Looks like a palm tree.
That's
Does
not
look like
a-
a palm tree.
It's deceptively like a crinoid, but
Looks exactly like
it's got almost has 5 arms.
That's beautiful.
Yeah.
I don't know which pathies this is.
What do you think? You want a black
Yeah.
coral?
Yes.
Yeah.
Why not?
Okay.
I do that.
We're exploring.
You owe me something. I'm not
You-
sure what.
I do.
Steve, tell us which
Yeah.
pathies this is.
All right, go ahead and come wide there, please.
Well,
This will go in the front box there.
Yes, please.
now we've been told we all have a beer ration on the last night, so maybe you win my beer ration,
Oh,
Greg.
but then I won't be able to work. Even one.
I
Even
don't
one.
think the rations are very big.
I'll just go giving those rations away willy-nilly. Nice.
That is beautiful.
Does look like a palm tree.
It looks so much like a stalked crinoid. I think 9 times out of 10 you'd fly by that.
Yeah,
Looks
the
pretty
good
floaty.
stalked crinoids down here, we expect them to be yellow and to have 5 arms, so it was really very, very tricky till we got a good view.
That was a good eye.
I feel like I'm creeping forward a bit. Hopefully not. It's gonna go in the front box too?
Yes.
All right, you guys had enough pictures before, or did you want to zoom before I grab it?
Oh, let's just get one more in case it's a special thing. We haven't heard from Steve about her name, but
Roger that, go ahead.
and if it just as it bends away, oh, that's good.
You want
And we
a
can
even
see the
tighter one?
base. We can see where it's attached. Okay. One more grab like that.
Getting lots of good screen grabs.
Some little symbionts living on it. Okay, thank you.
Roger that, full wide, please. That works for me. Go ahead and push on in there a bit there, please.
Tough perspective.
All right, our little pom umbrella.
Nicely done.
Wow,
Incredible.
yeah, that's perfect.
Go wide there, please. All right, Jake.
Open out.
Roger that.
Wow.
You want to open it halfway?
Halfway.
Thank you. Thinking this guy's going to want to float out, so I
Yeah.
might
There's like little
see if we can pin the palm tree.
Wow.
So let's do this instead.
Hopefully it'll sink on in there.
Go ahead. Actually, sorry, change my mind on what I want to do. Go ahead and start to bump it close there.
Roger.
Timing is critical here.
That's good there. Okay, ready?
Yep.
Actually, one more bump close. Sorry.
Oh.
Not the way you want, Herman. Just kidding. This'll work.
Yay.
It's
and it
sinking.
All
Yeah.
right.
perfect.
Nice.
Hey, that was
Very nice. That's going to be sample NA124 182.
Copy that.
thanks, Jake.
182.
Yep.
Very nice collection.
Good job spotting that one, Greg.
Yeah, super impressive. If this area just to the right of the sub looks good for sediment coring, probably be a good idea before we start to.
Yeah, we're running out of room.
Yeah.
Let's do that, Renee.
Raj.
Maybe all that wall of rocks was blocking the sediment coming down.
Some sediment.
Yep, hopefully it's accumulated nice and thickly here.
Yeah.
That's a good point, yeah.
Could just be wishful thinking, but
Sorry?
could be wishful thinking, but
Could be.
So anywhere around here then, guys?
Yes, please. Let's try
And
a couple.
before we land,
Niskin.
we're going to want to go for that Niskin
Roger that.
Niskin.
whenever is good.
So right now we're at 2 meters off, 2.5.
Yeah.
3
that
meters, is
sounds
that
good.
too far?
I think that's perfect. That's around the same depth as we get in the morning. It's perfect.
Raj.
And this is going to be Niskin bottle number 2.
Roger that. Thanks, Jake.
Yep.
Oh, what's going on here? Niskin number 2, Raj. That's only 2, yep. All right. Let's try.
Hot.
there you go.
Perfect. Thank you. That's going to be sample NA124 183.
183.
All right, then anywhere around here you guys said?
Yes, please. Wherever you sense deep sediment.
Think deep sediment thoughts. It's on a bit of a slope. Interesting core. All right, did you want to zoom on the sediment before I go and muck it up?
Yeah, let's have a quick look.
Go ahead and
Thank
push
you.
on in there, please.
Some brittle stars buried in there.
Some burrows.
We haven't collected one, so if Lisa doesn't mind, we could core over one.
Roger that.
We're taking 5 if possible. See what we can do.
Is that a worm on the lower right?
Oh yeah, scale worm.
Scale worm?
Oh, yeah.
All right, go ahead and come right there, please.
Ooh.
Coming out.
Nice scale worm. Or it could be a bomber worm on the bottom. We'll maybe have a look.
Ooh, bomber worm.
There's another bomber swimming away.
Yeah, those bombers will settle down to have a feed, and they'll take off.
Cool.
So am I correct in assuming that number 9, 10, 11, 7, and 8 haven't been taken yet?
Yes, that would be correct.
All right.
Super rubber band.
Oh my goodness. All right. And you said you want me to center one up on a brittle star?
Yes, we could. That would be a good one to try. Thank you.
Go ahead and push on in there, please, Jonathan. Roger that.
Yeah, starting to get demanding with our push core.
Go ahead and come wide, please. Thank you, that's good there.
And we'll
Little
get the
more.
squat lobster.
Okay.
Oh, you got
Got
the
that
brittle
brittle
star.
star.
you-
Go, 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 you got it. Look at that.
It almost looks like the worm in the middle left of the screen is bioluminescing red, or is that just a shine?
Full wide, please.
Usually with our lights, you get a bit of the reflectance rather than the bioluminescence. I don't think typically we would pick up. Iridescent. The ctenophores also kind of look that way too.
Right.
So you're going to assign sample numbers after they come in. Yeah.
They usually come in a different order.
Which push core is this?
Number 9.
We assign it we just made a note on number 9 that there's a brittle star in it. They'll usually assign sample numbers as they come in, just as they usually come in a little bit of a different order.
Any other organism you guys want in this next one?
No, thanks.
Reg.
Wherever you want, please.
Get down out of the brow bar there.
The rockfish to the left.
All the bacteria.
This might be up in the brow a little bit there. There. To check the light again. Go ahead and push on in there a bit, please. That's great. And come wide there, please. That's great. Pull wide, please. I'll do these 3 here, and then I'll go for this. I'll put them away and go for the second row, if that's all right with you guys.
Yes, no problem.
Jake, you want to do a little bump out on the toolbox there?
Yeah, got it.
Small bump.
A little bit more.
A little more. Yeah, that should be good. Gives me a little more room. Thank you. All right, Jonathan, go ahead and push on in, please. That's great there. Go ahead and come a little wide. Thank you. All right. Go ahead and get the brittle star one first.
Okay, that's push core number 9.
Go ahead and push on in there a bit, please. Thank you. Oh. Wanted you to close. All right. Go ahead and come wide there, please. At a weird angle. Sorry.
Hi guys, this is Kira in the lab. Just curious if we've taken another rock sample, if I missed it.
No, we would not do that, Kira.
Okay, no problem at all. Just checking.
Yeah, we're getting to the end of the sediment and we'll be on some more rock soon and then we'll check in.
Awesome. We didn't see any seep features, did we?
No, alas no.
Oh, okay. Cool. Carry on. Thank you.
Okay.
Not getting my alignment right on this guy. There we go.
Oh yeah, there you go. Nice.
Oh, hello.
Awesome. That's going to be sample number NA124-184.
Go ahead and push on in there, please, Jonathan. Great, thank you. Pull wide, Jonathan.
This is push core number 10. Beautiful.
All right,
Perfect.
number 11.
And just confirming push core number 10 was sample NA124-185.
Copy.
Go ahead and push on there again, please, Jonathan. Thank you. Pull wide, please. Ooh, this one doesn't have that much in it.
Oh, hello.
It's a good, it'll work. It's at least 10 centimeters, so they'll be happy with that. Thank you.
Roger that.
It's going to be some sticky mud.
It looks like quicksand.
definitely not quicksand when you're trying to wash out these tubes.
More like stuck sand.
Yeah. Clay.
But it's been really interesting to see the texture of the different sediments we've gotten at each of our dive sites. They're all a little bit different, which could mean you could maybe hypothesize or make a guess that the microbial communities and even the meiofauna communities could be different, but we'll have to see.
All right.
Beautiful.
So now on to 8 and 7.
Yes, please.
Roger that.
Just confirming push core number 11, sample number was NA124-186.
Copy.
Rennie, bubbles.
Bubbles.
Oh. They're strong rubber bands. That is one crooked push core now. There's a bunch of extra space in this one again, much like the last one. I chose a bad spot.
Just unlucky. Bull, will that be enough, do you think, for you guys?
Well, I think it's okay, yeah, because we can use the shorter ones and we'll give the longer ones to Lisa.
Oh, there
And
it goes.
and it goes. Oof.
Sorry.
Buried. No.
No problem.
Buried in. It's deep sediment.
I think this one might be like the last one where it was relatively, looks deep, but it's full wide, please.
It compacts down.
Yeah, or there's a lot of like dead space at the top of the last one. Not what you want. Where's it doing? If you want, the fullest ones are kind of to the right. I can hit this spot up over here,
Yeah,
kind of in the middle.
Good idea.
Roger that.
You can see the sediment pushed down in the middle with the core. It's compressing as it goes in.
All right. Actually, I suppose I want this one. Sorry.
It's going to be push core number 7.
Yeah, it really compacts itself. Full wide there, please.
Awesome. Push core number 7 is sample number NA124-187.
Copy that.
So, and then, so I'm just about. Yeah, this one's almost halfway full.
Yeah, even though you pushed it all the way in.
Yeah, full wide there, please. Oh. Jake, you want to bump out the toolbox a little?
Yep.
Good. That's good there. Thank you.
Roger.
Excellent work, Jess.
Amazing.
Thanks, guys.
These look great.
All right.
And just confirming that last sample, which is push core number 8, is sample number NA124-188.
Copy.
That's all of our push cores.
Well done.
Hopefully number 8 has enough in there for you guys.
Plenty. Those definitely all looked over 10 centimeters. And it's really interesting to note how that sediment compacts down. So I'm sure there'll be some interesting things to look at.
It's usually pretty gelatinous until you hit that stiff layer.
Jelly.
Onward, upslope.
Yes, please. Let's see what this feature up ahead has for us.
See if we can get there.
I'm going to get myself a little car wash while we're on our way.
Bridge nav. (100-meter step bearing 050. Thank you.
Making some filter feeders very happy.
Those worms are all going to come back.
So Greg and Paul, I'm headed towards this area right here where the contours are really tight on the off chance that that's real. This whole mound is definitely real, but if this is real, then that'll be quite a wall.
Yes.
Pretty cool.
Good idea. Looks fascinating.
Yeah.
It's going to be
Coming around to 050.
Roger that, thank you.
It's going to be an octocoral garden.
Yeah.
Lots of benthodites.
So in the rest of this watch, we want to collect 2 more rocks or 3 more rocks? What are you guys thinking?
3 more, please, if we can.
3 more rocks, Raj.
And a couple of slurps if possible. What is that
What
thing?
is that?
Whoa.
The long, tall guy?
Yeah, I don't think it's alive.
Go ahead and push on in there partially.
There's
Yeah,
More of those corals.
I think it's once was a stalked sponge, and there's another.
There's a black coral on the
Black coral.
lower end of that stalk.
Yeah,
It's umbellapathes.
Yeah.
Telepathy.
we got a beautiful one earlier.
Oh, and I
so
think there's an octopus
There's
on
an
the upper
octopus,
right.
Yeah.
oh yeah.
Yeah.
Oh, yeah.
Oh, octopus.
Yes.
Couple of books.
Look at them.
Possibly Alvin plate.
Yeah,
Octopus
another
alert.
black coral.
Possibly happen.
Steve, you called that an umbellapathes?
All right, go ahead and push on in a bit.
Umbellapathes.
Umbellapathes. Did you see the one we collected a little earlier?
I did not. I was a little distracted, but yeah, we've seen a few so far.
We got a really big, spectacular one.
Oh, look at that. Very cool.
Hey,
Go ahead and come wide. What is this transparent friend over here?
That is a predatory sea cucumber
Oh,
called Megalodicopia,
interesting.
Oh, wow.
which is not a filter feeder, it's a predator.
Wow, I would have thought that that was a tunicate.
Yeah.
Tempted to grab it.
What does it feed on?